Everybody else should probably mute.
Can you see that now?
Yes, we can.
Okay, so first of all, let me say how honored I am that you asked me to come and talk to you. It's been a long time since I've been with all of you, almost 20 years now. But I don't forget what a pleasure it was to interact with all of you individually during that time, and how much the cross-fertilization between the different groups meant to me to help our work go better. Unfortunately, now that I'm working on spinal cord, this spinal cord group has an annual meeting the day before neuroscience, the same time as the STG mini meeting, and so I haven't been able to attend that. So I'm particularly pleased that you're setting this up online so I can start to come to these meetings on a regular basis, because I'll always learn something new and interesting from you. So I'm just going to talk about a part of the work we did, which is the cellular and molecular mechanisms of amine modulation in the pyloric network. So Paul, I apologize, I'm not going to talk about the GPR cells, so if you want to log off, you can. But basically, I started working on neuromodulation in 1978 when I was a postdoc with Ed Kravitz, looking at the mechanisms by which serotonin and octopamine can modulate posture in lobsters associated with aggressive and passive postural states. And I really wanted to get into understanding what the cellular mechanisms were by which octopamine and serotonin did that. And to do this, of course, you need to have a network where you can look at the network, you can look at the cells, and you can look at the connections, and then you can see how serotonin and octopamine modified this. And I just basically ran into a wall. And I can remember, Eve helped me out, I think it was 1982, when you and I both gave back-to-back talks at the Society for Neuroscience meeting, 10-minute talks. And I talked about monoamine modulation of posture, and then you were talking about the STG. I think you were talking about transmitters identification in the STG.
Probably.
But this was the picture. Can you see my arrow moving around, by the way?
Yes.
Good. This was the picture that blew my mind. Here's a network where you knew all the cells, we knew virtually all the connections, and it gave an interesting behavior, and it was tractable. And so I went to Eve after her talk and told her how amazed I was and how do I get started on this. And she very, very generously invited me and my graduate student, Bob Flam, to come to Brandeis for a week, where she and Judith Eisen taught me the prep, taught me how to do the dissection, and we started a project. I've been working on serotonin, so we chose serotonin modulation in 3 different species of crustacea. And we roped Barb Belts into the project to do some immuno, and we published a very nice story. So I'm extraordinarily grateful to Eve for your generosity in welcoming me, showing me the way, and helping me get started in this, and having this great community to work with. So most of you work on crabs now, but we worked on Panularis the whole time, and there are a couple reasons for that. First of all, if I was going to study cellular and synaptic mechanisms, we need a circuit, and the early network circuit breaking had been completed in Panularis and had not yet been started in the crab. Secondly, there's a lot less electrical coupling within the pyloric network, which makes it easier if you want to isolate neurons, which I'll talk about in a minute. Third, the neurons are bigger, and it's relatively straightforward to do 2 electrode voltage clamp, which is much better than a single electrode switch voltage clamping. And then finally, in Panularis, the pyloric rhythm is completely dependent on STN modulatory input. So it essentially always stops when you block the STN with either sucrose or TTX. And so you can really get the system in a stable baseline pattern, and pretty much every single day. So this is the situation. Basically, we take a system that's either already cycling like here, or that's silent with a TTX block, and you add dopamine or dopamine or serotonin. Let's see, someone is not muted. I'm not sure who it is, but if you could all check to make sure you're muted, that'll make life a little better, I think. Basically, what you see is you see changes in the motor pattern, as all of you know. And basically, dopamine and octopamine and almost every modulator you can think of will turn on a silent motor pattern and will modify an ongoing one. And while this looks like a fairly subtle change, pretty much every parameter that can be changed is changed. You change the cycle frequency, which in this case slows down a little bit. You change which neurons are active, where some neurons become more active and some less active. You change the intensity of activity when the cells are firing, and you change the phasing, where the PD is phase delayed and sometimes silenced, and the LP and PYs are dramatically phase advanced. And I want you to remember the phase advance of the PY for something I'll talk about later. And of course, you know all this already. This is not limited to dopamine. We worked on dopamine, serotonin, and octopamine, although mostly we worked on dopamine. And you get different patterns with different monoamines, and you get different patterns with the peptides, as many, many of you have studied. So the question then that I was interested in was, how do these monoamines do that? What are the mechanisms by which they reconfigure these neural networks so that you get a different output out? Because this kind of sounded to me like a great model for understanding flexibility of behavior in all nervous systems, and would therefore be kind of a generally interesting question that's not specific to the STG. So the way that it can happen is pretty much the ways that you think it might happen, and these are the two things that I'm going to talk about today. Dopamine and the other amines can either change the properties of the individual neurons, their intrinsic firing properties, by changing ionic currents, or they can do so by changing the strengths of all the synapses. So this figure here kind of summarizes the talk I'm going to give today of the cells that are in some way or other excited by dopamine or inhibited, the synapses that are strengthened by dopamine or weakened by dopamine. So how do you actually show this? We did this, first of all, looking at the intrinsic firing properties of the neurons. And to do this, the easiest way to us, it seemed, to do this is to try to study each neuron by itself without the distractions of all the synaptic inputs from the other neurons. And so we had to figure out a way to do synaptic isolation of these neurons. And John Miller had already been doing this, and so we followed his direction. So basically, here's an example of isolation of a VD neuron. The VD neuron gets modulatory input through the stomatogastric nerve, which can be blocked with a sucrose block or TTX. It's electrically coupled to the ABs and PDs, which can be removed by killing them with 5,6-carboxyfluorescein photoinactivation. And then it receives a couple glutamatergic synapses, which can be blocked by adding picotoxin that blocks the glutamate synapses. So instead of having this rather complicated pattern here, you have the VD totally isolated, receiving no synaptic input. And in this particular case, the PD fires tonically. Then to this isolated neuron, you can add dopamine. You can study how dopamine modifies its firing properties, the currents that are affected using voltage clamp. You can use calcium imaging to look at calcium. And you can also isolate pairs of neurons. For example, if you wanted to isolate the LP-VD pair, you just photoinactivate the IC and you'd have that pair isolated. So Bob Flam, my first graduate student, did this for all the cells. And this is the results of a lot of work that he and I did together, where under control conditions, the neurons show varying baseline activity. Some of them are silent, some of them are tonically firing, some of them are a little irregular. The AB is usually silent. But then you can add dopamine or octopamine or serotonin, and what you get are different patterns of activation and inhibition. So the take-home messages are three from this. First, that all the neurons are targets of neuromodulation, not just one and not just a few. Secondly, that the effects of an amine vary on different neurons. So dopamine, for example, makes the AB neuron oscillate. It inhibits and hyperpolarizes the PD and BD and excites the LP, PY, and IC. And although it's not obvious here, these are neurons that are in the up state of a bistable state. They're actually bistable neurons that stick in the high state unless you apply a bias current. Octopamine activates all the cells weakly or strongly. Serotonin activates some cells, inhibits some cells, and has no effect on a couple of cells. So we can then, in a kind of reductionist way, go one step further and say, how do they do that? You know, let's say this AB cell, it becomes an oscillator with dopamine. Seems to become a different kind of oscillator with octopamine, and not so obvious, but possibly different even with serotonin. What are the mechanisms by which dopamine affects this? So the mechanism for this is dopamine and the monoamines always act on G protein coupled receptors in crustacea, and therefore they activate second messenger pathways, which can alter the properties of ion channels, which shape the neuron's firing properties. So to do this, we did a whole bunch of 2-electrode voltage clamp. And I know this is not of general interest to most of you, so I'll try to go over it relatively quickly. But many components of these currents can be modified by these monoamines. The obvious one is just to increase or decrease the amount of current there is, and that's what most of our models of alteration of these neural networks do. They just increase or decrease the maximal conductance. But that is certainly not all that these monoamines do. They can alter the voltage dependence. For example, they can alter the V1/2 for activation or inactivation. Sorry, I keep pushing the button wrong. They can actually change the degree of dependence of voltage to voltage, that is the slope of the voltage curves. They can change the kinetics of ion channel enclosing. They can alter regulation by intracellular calcium, and in addition, they can alter currents which are not voltage dependent, leak currents, either by increases or decreases. And in fact, all of these do occur during dopamine modulation. So let me just give you one example, if you will bear with me. This is dopamine modulation of the transient potassium current in PY neurons. I showed you earlier that when dopamine is added, the PY neuron shows a marked phase advance and fires earlier relative to the onset of the AB neuron. And part of this is due to changes in synaptic drive inhibiting the PY, but a significant fraction of it is also intrinsic to the PY neurons themselves. And you can show this using isolated PY neurons, and instead of having synaptic inhibition, you just hyperpolarize the cell to mimic the synaptic inhibition and then depolarize it just above threshold. And what you see is it recovers from inhibition and then starts firing, but the more you hyperpolarize it, the longer this delay is before it starts firing. So this will determine the phase of when this cell starts firing in an isolated cell model. When you add dopamine, you can see its phase advance, just like it is in the intact network. It has a much, much shorter delay to fire, and the slope is steeper when it does. This actually has a number of causes, and I'm only going to talk about one here, which is the transient potassium current that's seen here in voltage clamp. This is an outward potassium current that, like the voltage-sensitive sodium channel, has both a voltage activation curve and a voltage inactivation curve. At rest, it's mostly inactivated. So you can see here, there's very little activation at the normal resting potential. But if you hyperpolarize it, what you're doing is moving up this inactivation curve to relieve inactivation and to de-inactivate the channel. And then when you depolarize the cell, you jump over to the activation curve and the channel opens. But once the channel opens, it also starts to inactivate, so you get this transient current that rises to a peak and then falls down. Now, all this happens much more quickly in dopamine, so the hypothesis is that there's less of this current when you add dopamine. And when we did the voltage clamp studies, that's in fact what we found. And in fact, when you do Boltzmann analysis on these data, you find that the maximal current is reduced by about 30%. But that's not all that happens. Another thing that's kind of obvious here is that the voltage activation curves are shifted over by about 10 millivolts. So it actually takes more depolarization to get the channel to start opening. So in the range where the cell's oscillating, you will actually have less of this current activated because the voltage activation curve is shifted over, and not simply because there's fewer channels. And then finally, there's a change in the kinetics, where if you look at the inactivation of the channel, it appears to inactivate much more rapidly. This is kind of obvious here, especially compared to the washout. So our first hypothesis is that it actually accelerates the time constant for inactivation in this channel. Turns out that wasn't correct. When we did fits to the exponentials for this inactivation, it turned out it could only be fit by 2 exponentials. One, a rapidly inactivating component with a tau of around 20 milliseconds, and the other, a slowly inactivating component with a tau of around 100 milliseconds. At that time, we didn't know what caused that, but later, Deb Barrow, while she was in my lab, cloned the gene that encodes this channel. It's called SHAL, or KV4, and in lobsters, it undergoes a tremendous amount of alternative splicing. And the splice variants vary in things like their voltage dependence for activation and their rate of inactivation. So this means that these PY neurons have, at a minimum, 2 splice forms and probably many other splice forms that lead to 2 average time constants for activation and inactivation. What you can see here is that dopamine does not alter the taus, the time constants themselves. So it isn't that it's inactivating more rapidly. What's happening is that the relative amount of the rapidly inactivating component and the slowly inactivating component is changed. So there's no change in the rapidly inactivating component, but a decrease in the slowly inactivating component. And that washes out. So there's, of these various splice variants, one of them appears to be insensitive to dopamine and the other is blocked by dopamine. We don't fully understand what causes that. So the consequences of all this is that the cell shows more rapid post-inhibitory rebound and is phase advanced. There's less potassium current, it's harder to activate it with voltage, and it inactivates more rapidly. So it's not simply a decrease in conductance. So I realize that was boring for most of you. I could show you dozens of slides like this, but I'll just show you the summary, which is this slide that Eve doesn't like very much, except it does summarize what we've done. And this is not a complete list because at some point we gave up and stopped looking, but you can see that each neuron, when we just look at dopamine alone, there are multiple ionic currents that are affected by dopamine. There isn't just one current affected. And I really don't understand why this is true with all of the monoamines and it's not true with the peptides. That's a real mystery to me. And it'd be fun if we could talk about that maybe afterwards. So you can see some of the currents are enhanced, some of them are decreased. Let me just say these are functional enhancements. I'm not saying these are increases in the amount of current. For example, this increase in the hyperpolarization activated current in the LP, there's no change in conductance at all, but there's a 20 millivolt shift in its voltage activation curve. So at the normal voltage range that the cell is in, there's much more of this current activated because of the voltage shift, and that's a cyclic AMP dependent voltage shift. But each neuron is affected differently, and they're all affected in different ways. This looks pretty complicated. In fact, it is complicated. So let's talk about two specific complications that show the complexity. The first is something that is, of course, very familiar to all of you, is that you can get similar outcomes by independent mechanisms. Astrid's beautiful modeling work and Jorge's spectacular work on the many, many different amplitudes of currents in the LP cell that all fire the same way show this. And it turns out the neuromodulators can evoke similar outcomes by completely independent mechanisms too. And the example here is in the AB cell, where if you isolate the AB cell, as I said, it usually falls completely silent. But then you can add dopamine and serotonin and it resumes firing. And to a first approximation, it looks pretty much the same kind of oscillations. If you do this enough and you do statistics, the dopamine oscillations are a little bit larger, They're a little bit faster. They fire a little bit higher frequency, but there's a really small change. I did all these experiments, and if you were to take 10 days worth of dopamine and serotonin recordings like this and mix them all up and ask me to sort them into a dopamine pile and a serotonin pile, I couldn't do it. They're too similar to do it by eye. Nonetheless, these mechanisms of bursting have absolutely nothing to do with one another, completely independent. And back in the 1980s, when we started this, we could only do simple experiments, but these simple experiments showed this. The dopamine bursting, for example, is absolutely dependent on calcium. If you reduce the extracellular calcium to 25%, you block the bursting in dopamine completely, whereas serotonin continues oscillating very nicely. In contrast, the serotonin-induced bursting absolutely depends on voltage-dependent potassium channel or sodium channels. If you add TTX, the dopamine-induced oscillations continue just with the action potentials removed, whereas the serotonin oscillations are completely abolished. Later, Lola Kadiri, who many of you knew as Lola Muhammadiyeva, used more sophisticated methods to show these differences. She used two-photon calcium imaging, in this case using Indo-1. So she would fill the isolated AB cell with Indo-1 and image it with two-photon, add TTX to block it, and you can see a decrease in the calcium signal as it stops oscillating, and then add dopamine, it starts oscillating again. And what she found is that the first thing that happens is the release of calcium from intracellular stores. These are measurements from different varicosities in the AB neuron. You can see that when dopamine is added, one of the early things that happens is an increase in intracellular calcium. And this is well before the cell starts to oscillate. And eventually, when it is oscillating, the calcium's oscillating right along with it. But before then, it rises early. And this is from intracellular stores, because if you block the release of calcium from the endoplasmic reticulum with ranitidine, blocks the ranitidine receptor, and zestospongin that blocks the IP3 receptor, you block this calcium increase. The cell still depolarizes because dopamine does many things to the cell, but it won't oscillate. And if it is oscillating and you clamp intracellular calcium, it stops oscillating. If it is oscillating, you block the ranitidine receptor to block release from intracellular stores, it stops oscillating. The consequence of this increase in intracellular calcium is to activate the calcium-activated non-selective channel. And if this is not done, in dopamine, this cell will not oscillate. Flufenamic acid is a blocker of the CAN current, and it also abolishes oscillations. This is a super, super complicated voltage clamp, and I'm not going to try to explain it except just to say that this curve here represents the inward current carried by calcium across voltage-dependent surface calcium channels. And when dopamine is added, it changes a bunch of outward currents, so the baseline is shifted, but this current is much, much decreased. There's actually a significant decrease in voltage-dependent calcium entry across the membrane. This part represents the calcium-activated CAN current, and you can see it's very, very dramatically enhanced by dopamine. So the center of the oscillation induced by dopamine is an increase in intracellular calcium, which then triggers, this is by release from the endoplasmic reticulum and intracellular stores, activates the CAN current. These are the essential components. There's a whole bunch of other things that dopamine does, none of which are essential, but all of which help. For example, there's a reduction in leak current that helps the depolarization. Enhancement of the H current adds to the depolarization, as I said, there's this paradoxical decrease in the voltage-dependent current. During the burst, there's an increase in IKV, which at first seems paradoxical, but may help the cell fire spikes at higher frequency by causing more rapid repolarization. And then it has to be terminated. There's enhancement in IKCA, probably because of the enhancement in intracellular calcium. So this really depends on calcium. Serotonin doesn't, completely unimportant. It depends instead on a persistent sodium current called INAP. This is a component of the normal sodium channels which doesn't inactivate. It's blocked by TTX. It's also blocked by low concentrations of ribosol, which abolish bursting. And what happens is that the slope of the rise to the burst gets flatter and flatter until it finally stops. And I'm going to skip almost all of this, but basically serotonin itself does not modulate the persistent sodium current. What it does is downregulate opposing currents, okay, in particular the lead current and IKCA, so that the ratio, say, of the persistent sodium current to the lead current goes up. And so the net result is that you can get a steeper rise to the point where the cell starts to fire. So this, just to complete this, here's a cell that, an AB neuron that's caused to start firing with serotonin. We abolish it with ribosol. In the continued presence of ribosol, we add dopamine, and by a completely unrelated mechanism, it starts oscillating, and the oscillations look almost identical. So two completely different ways to get there from here. Again, this was surprising to us at the time. It's no longer surprising because of the wonderful work that all of you have done since then. The other thing that's complex is that when you look at that figure, you find a lot of cases where it looks like the monoamines do the wrong thing, that would oppose the effects. And these are circled here. Where, let me take a couple, I just talked about this one where the AB cell oscillates by a calcium-dependent mechanism, but it decreases the voltage-dependent calcium current, and it's a lot. It's about a 70% reduction. It's a big, big effect, and Bruce Johnson showed this first. In the VD cell, the cell is hyperpolarized and falls silent, and we don't know the mechanism for that, but one thing we found was that it enhances this hyperpolarization activated inward current, which is going to be activated as the cell hyperpolarizes, and we'll try to depolarize it, right? So there's other examples. And so these could be evolutionary mistakes, but they also could be some way to kind of constrain the degree of modulation of the cell to keep them from becoming overmodulated and falling out of the dynamic range in which they're able to do their job properly. And I'll have more to say about that when we talk about synapses, which I'm now going to talk about. So the other way that you can reconfigure a neural network like this is by changing the strengths of the synapses. And again, this little cartoon, the thick lines are synapses that are functionally strengthened, and the dotted lines are synapses that are functionally weakened. And Bruce Johnson did virtually all the experiments that I'm going to talk about here with help from Jack Peck and several other people. And the way that he did this was, instead of isolating single neurons, he would isolate single pairs of neurons connected by a synapse. And then he could add the monoamines, and he could see what happens to that synaptic connection. And what he studied primarily was graded synaptic transmission, since we think that graded synaptic transmission plays a major role in the pyloric network. So these are examples of his work. Here are all the synaptic inputs onto LP neurons, okay? And what you see is that there are many, many different effects of dopamine on different synapses. There isn't just one response to dopamine. So the AB to LP synapse is actually strengthened when you add dopamine. In contrast, the VD to LP synapse is weakened by dopamine. The PD to LP synapse is actually completely abolished. It doesn't matter how much you depolarize the PD cell, you don't see any response at all. And this is despite the fact that the postsynaptic cell is still sensitive to acetylcholine, the PD neurotransmitter. So this must be a presynaptic effect. You also have synapses that when you remove the neuromodulators, these synapses fall silent. So the PY to LP chemical synapse, for example, is abolished when you wash out the neuromodulators. But if you add dopamine back, it recovers and it's present. You wash out the dopamine, disappears again. You can alter electrical coupling, and we're not exactly sure in most cases exactly how that happens. There could be a change in the junctional conductances, or it could also be due to changes in the input resistance of the cells. And in a couple very interesting cases, there's actually sign reversal of the synaptic connection by dopamine. This is one of the examples that Bruce found from the LP to the PY, which has a rectifying electrical synapse and also a chemical glutamatergic inhibitory synapse. When all the neuromodulators are washed out with a block on the STN, the chemical synapse falls silent, but the electrical synapse is active. And so the net consequences of depolarizing the LP are to depolarize the PY. When you add dopamine, this electrical synapse falls silent and becomes undetectable, but the chemical synapse is activated, and so now you get a hyperpolarization response to depolarization of the LP cell. So you get sign reversal. And there's several other synapses that have mixed components like this where we saw this sign reversal effect. Now, again, being reductionistic, we wanted to understand how these changes in synaptic strength occurred. And obviously they could occur presynaptically by the nerve terminals releasing more or less neurotransmitter, or postsynaptically by the cell becoming more or less sensitive to that transmitter or changing its input resistance. So we looked at these. Presynaptically, this work was done by Peter Kloppenborg, and what he did was use 2-photon calcium imaging, in this case I think it was calcium green, to look at calcium, voltage-dependent calcium entry into single varicosities from a single isolated cell. So he synaptically isolated a neuron, he would inject the calcium green into the cell, voltage clamp it, and then step it from -60 to 0 to activate voltage-sensitive calcium channels and look for the increase in calcium. And the idea is if there are changes in calcium entry, this could cause changes in the release of neurotransmitter from those terminals. And so Peter did this, for example, for the PD neuron. And what he found is that about half the varicosities that he looked at, the net effect was to have decreased calcium coming in. These are the F over FO plot. It's cut down by about 50%. And then it recovers with time. So this is the case where you get decreased voltage sensitive calcium entry into the terminal, which presumably would decrease release, especially since, if you remember from your introductory neuro, release is related to calcium raised to the 4th power. So a 50% reduction in calcium is a big, big, big reduction in release of neurotransmitter. On the other hand, you can have postsynaptic changes in response to the neurotransmitter. So Bruce did these experiments where he just threw away the presynaptic cell and replaced it with a pipette, in this case, glutamate. So he could monitor the effects of the glutamatergic neurons onto their postsynaptic cells just by ionophoresing glutamate onto it. And here's a case of the PD neuron that has a response to glutamate. Until you add dopamine, that response gets much, much smaller. And then it washes out as you recover from dopamine. So this is a postsynaptic change that would weaken the synapse. So this is another one of the slides that Eve doesn't like because it's so complicated. The problem is that it is complicated, that the effects of these monoamines on synapses are not simple. Pretty much any component you can imagine is changed, and it's changed in pretty much every synapse. So let me just walk you through this. The little pluses and minuses in the nerve terminals show the effects of dopamine on calcium accumulation, enhanced or decreased. The little pipettes show the response to glutamate ionophoresis, enhanced response, decreased response. And then also the input resistance of the cell will generically increase or decrease its responses to all synaptic inputs, and so that those can go up or they can go down. So this is really complicated. So let's talk about 4 components of pyloric synapses, and these are the last things I'm going to talk about. The first is that we frequently find opposing pre and postsynaptic actions. If we go back here, some of these work very nicely. So if you look at the LP to PY synapse, This synapse is strengthened by dopamine. If we look at release of transmitter from the presynaptic cell, release of the increase in intracellular calcium, it's significantly increased by dopamine, and that presumably causes more transmitter to be released. If we look at the response of the postsynaptic cell to glutamate, it's enhanced. If we look at the input resistance, it's enhanced. And this is the example I just showed you, that the opposing rectifying electrical synapse that would oppose the hyperpolarization is weak, in fact, is abolished. So everything works in the right direction for LP to PY. For this one, not so obvious. So let's take a look at that. This is the LP to PD synapse. Now I'm showing you results. We work with Farzan. Bruce worked with Farzan to make averages of the oscillations of the LP cell, and then voltage clamp the LC and drive it with this artificial oscillations, and then record the postsynaptic response so we can see the peak response, then the decay of the synapse as it shows weakening over time. But you can see that for this graded synaptic transmission, dopamine enhances this synapse. It enhances it for the first step, it enhances the steady state level. This synapse is strengthened. If you look presynaptically in the nerve terminals of the presynaptic cell, the LP cell with calcium imaging, you see that dopamine enhances the calcium entry. So presumably enhances the release of transmitter. If you look postsynaptically, I actually already showed you this figure, there's a dramatic decrease in responsiveness to glutamate, the LP's transmitter. So we have opposite effects, more transmitter coming out and less response to the transmitter. The net effect is the synapse is strengthened. So clearly the presynaptic enhanced release outweighs the postsynaptic reduction in sensitivity to the transmitter. This is not rare. Here's a bunch of other examples where the same thing happens, where typically the result is enhanced transmitter release outweighs reduced transmitter responsiveness. And again, you might wonder why this would happen. And Bruce and I argued in a review article that, again, these opposing effects of neuromodulators are there to constrain the degree of modulation that is allowed to prevent the system from becoming overmodulated and falling out of the dynamic range within which it can function. And so it's kind of like you have an accelerator that pushes the system in one way and a brake to constrain it from being pushed too far. So it stays in a certain sweet spot within which the network is still functional. So that's the first of complexities. The second complexity we found is that there are highly variable actions of these neuromodulators on calcium entry at the terminals of a single neuron. And this is work done by Peter Kloppenborg, where he looked at PD neuron. I showed you earlier that when you add the PD neuron, its synaptic output is weakened. The net effect is a reduction in calcium entry into the cell, and that's true for about half of all the varicosities. The other half actually show no change at all. And we were surprised by that and didn't fully understand it. Later, Deb Barrow, working in her lab at Georgia State, provided us with an answer to that she cloned the dopamine receptors, and there's a D1 receptor, which is excitatory, and a D2 receptor that's inhibitory, that this enhances cyclic AMP and this reduces cyclic AMP. And she found that the PD neuron only expresses D2, and with some very elegant electron microscopy, she showed that these receptors are highly concentrated on varicosities, which are presumably nerve terminals or postsynaptic sites, but that only about 40% of the varicosities had any dopamine receptors. So those 40% presumably represent the numbers where the release of transmitter is reduced by the release of intracellular of calcium entry. The others, maybe there are no dopamine receptors there. We have no idea why only 40% of them do this. All the synaptic outputs from the PD neurons are reduced. In opposition, the LP neuron, on average, the output synapses are strengthened, and for about half the synapses, it goes up. The calcium entry goes up, but the other half, there's no change. And Deb, again, in her lab showed that only D1 receptors are found in the LP neuron. They didn't do the EM on the LP neuron, but if it's like the PD neuron, only half the varicosities actually have these receptors. An interesting contrast is in the PY neurons, where the synapses are all strengthened, but when we look at the individual varicosities, some aren't changed, some are strengthened, and some are weakened, okay? About 30, 30, 30, and this is all work done by Peter. At first we thought, well, this reflects different PY neurons. There are 8 PY neurons, and maybe some of them show an enhanced calcium entry in terminals, and some of them show a reduced. But Peter showed that that wasn't true. He found many examples of varicosities right next to one another in a single cell, one of which showed an enhanced calcium entry to voltage, and one of which showed a reduced calcium entry to voltage. I don't think Deb has done the analyses, but we would assume that PY neurons express both D1 and D2 receptors that are differentially expressed on different nerve terminals. And why they're differentially expressed, I don't know. And that is a question for the future. A third complexity is that you can actually get opposing effects on graded and spike evoked release of transmitter. So we normally just think about the graded transmitter that makes these cells oscillate, but of course we always see the spike evoke component as a small part of it. Here from the LP, you can see the IPSPs, and the LP itself is terminated by very large spike evoked IPSPs from the PY neurons. It turns out these can be differentially affected by monoamines. So if we go back to the synapse I've talked about many times, the LP to PD synapse, the graded transmission is enhanced, as I just showed you here. And here's another example using square pulses, where under control conditions, you get a certain IPSP, and with dopamine, you get a much, much larger IPSP. Amir Ayali then looked at what happened to spike evoke transmission. So he very, very briefly depolarized the LP with a current step, or else depolarized it right above threshold, right near threshold. So occasionally it would fire a single action potential. So you could see spike evoke transmitter release. And what you see in this case is that the response is weakened. It's not strengthened, it's weakened. And this also happens at output terminals from the VD cell and so on. So this is not unique to the synapse. I went through the argument that the strengthening of the graded synaptic transmission must be presynaptic, because the postsynaptic cell becomes less responsive, and yet you're getting a bigger response. For the spike evoked response, it turns out that the degree of release is exactly predicted by the postsynaptic reduction in input resistance. So the PD cell gets enhanced leak currents, it becomes more leaky, and therefore shows a smaller response. So this is a purely postsynaptic response, the reduction of spike evoked transmission, whereas an enhanced graded transmission is a presynaptic response. Why this is not affected by reduced calcium entry is not clear, and we never solved that problem. Okay, the final thing is to talk about the dynamics of synaptic depression. Farzan and others have looked at synaptic depression a lot and shown how important synaptic depression is for normal functioning of these neural networks. And so Bruce with Mark Kavarta studied this, and we did it in 2 different ways. This is an example of the PD to LP synapse, where we can either give square pulses, in which case you get a peak response that then decays and shows synaptic depression to a steady response. Or you can use averaged waveforms and voltage clamp drive oscillations in the presynaptic cell and look at the responses in the postsynaptic cell. And again, the first oscillation causes the biggest peak, and it decays to a steady state after 5 or 10 oscillations. You can measure a depression index, which is basically the percent drop from the peak to the steady state. And you can also measure the time constants of the rate of change from the peak to the steady state. You can fit that with an exponential and get the depression, the tau depression. And you can also give a series of steps and then give a step at varying times afterwards and look at the recovery. And so you can get a tau for the recovery from depression. Turns out all of these are subject to neuromodulation. So looking at the same synapse, say with octopamine, So octopamine strengthens this synapse. You can see it here, both the steady state and the peak are enhanced. And again, with waveforms instead, you get a larger initial response and a larger steady state response. Furthermore, it decreases the amount of synaptic depression. So not only is the initial response larger, but it depresses less. So you get about a 15 to 20% reduction in the amount of synaptic depression that occurs. Furthermore, you change the time constants so that the rate of depression going into the depressed state is slowed down by octopamine. It has a longer tau, while the rate of recovery from depression is sped up. All of this will make this synapse stronger no matter what's happening with the oscillations and so on. So this all kind of fits. Serotonin, on the other hand, weakens this synapse, as you can see here, both the peak and the steady state for both square pulses and realistic waveforms. And in fact, it depresses to a greater extent with serotonin than under control. So in this case, it's more depression, and it's a faster onset of depression. All of this would make this synapse weaker and more likely to depress, but it also shows faster recovery, which we don't fully understand. And then finally, we fussed around with dopamine, and it turns out if you use lower concentrations of dopamine at this synapse, you can find a condition where dopamine doesn't actually change the strength of the synapse. Neither the peak nor the steady state are affected. The depression index is unaffected. All it does is change the rates of going into depression and coming out of depression. So the time constant for depression in this case is accelerated for both going into the depression and recovering from the depression. So this is actually the last paper we published, a data paper on the stomatogastric ganglion. We don't really know what this all means yet. And since we're working on mice, we're not going to know unless one of you picks it up. There are lots of possible mechanisms how this metamodulation could occur. And I'm not going to go through the details, but there's a lot of different ways that you can change the rate of depression and the rate of recovery from depression. Okay, so that's basically what I wanted to talk about today. I'm not going to go through these conclusions, but the take-home message is that network neuromodulation is very complex and involves many components. It's not a single component. With the monoamines, again, in marked contrast to the peptides, many ionic currents are involved, not just one. This makes perfectly good sense because the D1 dopamine receptor, for example, enhances cyclic AMP. And cyclic AMP, either by a direct action, as on the H current, or indirectly through phosphorylation, can modify the properties of hundreds of proteins, including many ion channels. And D2 decreases cyclic AMP will have a negative effect on those same hundreds of ion channels. So it's not surprising. What's surprising for me is that why this is not true for the peptides. Okay, so as all of you have to write whenever you write a grant proposal, this is what happens in crustacea is super exciting, but at some point it has to relate to vertebrates, or else the NIH won't give you the money. And so all of us write good explanations for why we're covering general principles that are generalizable. And for the last 20 years, we've actually been testing that and finding many similar things in the mouse spinal cord. So all the work you're doing, pardon me, all the work you're doing actually does transfer up to the other systems. So I just talked about a little bit of the work we've done, the work on modulation. We've done many other projects, and these are the people that worked on this, this project. Bob Flam really helped us get started. He's the first person to work with me when I first graduate student, did the first work on how many different effects there are. Bruce Johnson really is the leader of all the work that I was talking about on synaptic transmission. He's had many assistants, Jack Peck, Mark Kavarda, Lauren Schneider, and then a large number of people have worked on the intrinsic mechanisms, including Anne Jane Tierney, Lisa Coniglio, Ola, Paul's in there, and all the others, and I thank all of them. So that's all I have to say, and I'd be welcome to hear some conversation about it.
So I would like to go back to the issue about the amines and the peptides, because I think your work is so extraordinarily, seeing it all together is really quite wonderful. And by the way, it's not the work that I don't love. It's those slides. I don't love. It's hard to look at, but the work is beautiful. But what I think is worth thinking about is peptides are always found as co-transmitters. And so to the extent you might have 2 or 3 or 4 or 5 or more co-transmitters, which could have different actions, what the amines may be doing with one molecule, maybe the peptides and their, as part of a co-transmitter package, are doing to get those potentially opposing actions or balancing actions done. So that's the way I think about it, is that you've expanded the time course maybe, but by using the, if you put GABA and proctolin and CABTRP together, you're going to get a lot of, both synergistic or antagonistic effects. So I think that's probably the answer, if there's an answer, is that the peptides are never found alone.
Right. Well, it's also relatively rare that the amines are ever found alone. Certainly in the vertebrates, I don't think that's ever happens. Paul identified the GPR cells, and one of the first things he found is that they contain acetylcholine. In that case, what Paul showed, and you can correct me, this is old stuff for me, Paul, so I'm a little rusty, was that the acetylcholine was acting by rapid transmitter actions, nicotinic actions. And then a peptide was found in those GPR cells too. So we have a peptide, we have serotonin, we have acetylcholine acting by a nicotinic mechanism, all in the same cell. The receptors, I know of no instances where the receptors activated by a peptide are not G protein coupled receptors. Those G protein coupled receptors activate second messenger mechanisms, which are generic. Increases in cyclic AMP, changes in intracellular calcium, phosphoninositide metabolism, which then spread out and affect lots and lots and lots of things. So
I wasn't saying that. I was just saying.
But it's just it means the peptides shouldn't just affect one current. It just doesn't make any sense if they affect one current. If they're activating by increasing intracellular calcium or increasing cyclic AMP, these are going to affect hundreds of proteins in the cell, not one.
Remember
that's a question in my mind.
Well, first of all, it's not all the peptides, but also the second messenger activation by the IMI supposed set is very, it's been very thorny. So,
And also don't forget that there are lots of G protein coupled effects that are direct, not necessarily through a soluble second messenger. And I suspect the peptides in this system, we have no proof, but I suspect it might be much more directly linked to the receptor, to the channels than perhaps the amines are.
Right, so that makes things even worse rather than better, because then you're adding yet more possible targets, because then you have the direct effects as well as the very large number of indirect effects that are found everywhere else. No, Eve, you make a good argument for how you could get around this funny thing and get multiple mechanisms, but it doesn't solve this basic kind of cell biology question, that it's kind of unreasonable that a G protein coupled receptor will only activate one channel. Doesn't make any sense.
Ron, can I ask you a different question or address a different point that you made, and that is this overmodulation thing. We use that to argue why we have so many peptides, so many modulators and so on, but do we really have any evidence that that is actually what these multiple peptides are doing? I don't think so, and I think there is no program really to address this overmodulation thing directly. Maybe Dirk will disagree, but I mean, if a synapse is strongly activated and curves are shifted, how does that affect the effect of other modulators? To me, is a totally open question still, and difficult one to address, right? What are your thoughts?
Yeah, no, that's really important. Again, saying what Eve said, that these things are not found alone. And so everything I showed you, as complicated as it is highly oversimplified. And modulation of modulation is certain to happen. And so you might take a cell into a state with one neuromodulator where another neuromodulator will have an action that it wouldn't have without the first modulator. Or the opposite can happen. Something has to keep it from falling apart. And certainly there's these many homeostatic mechanisms that have been so elegantly demonstrated. This is one of the things I think that the STG really, one of the great gifts to neuroscience is our understanding of homeostatic compensation and modulation. But what keeps the system, this is something Eve and I have talked about for decades, and Eve really has pushed harder than anybody else, is how can you get flexibility and stability at the same time? And one way of having kind of this system of checks and balances that kind of hold the system in and don't let it go too far one way or the other. We don't have any proof of that. You're absolutely right. This is just an idea that has not been proven.
Well, to go back to something that Mikey would be saying if he weren't muted, and that is when we study the exogenous application of substances one by one, rather than studying them as they're released as co-transmitters from neurons, we are perforce finding conditions or states that are not necessarily physiological. So I mean, I know that he would argue that it's only when you really study the activation of the neurons that contain the co-transmitters, or the other thing that we've been all guilty of, not paying as good attention as we might to when each one of those peptide modulators acts, or amine modulators that's hormonal liberated, under which conditions each one of them is being liberated. And it could be that you never get hormonal liberation of peptide 1 at the same time as hormonal liberation of peptide 2 or 3. We study them as if they're all, potentially available to the system at the same time. But it could be that CCAP is only available under certain physiological conditions, and that RPCH is only available under different physiological conditions. And so that, we're possibly creating a red herring by not going back to the biology.
Right. No, I agree completely.
Biology, of course, the fun part.
Yeah, no, it's a very, very good point. Paul, as part of his thesis, did compare the effects of stimulating the GPRs to the effects of bath application of serotonin, found, correct me if I'm wrong, Paul, basically the same thing. So it wasn't that we were qualitatively wrong on anything by bath applying with regard to comparing, and that's the sole source of serotonin, by stimulating the GPRs or by bath applying serotonin, you got the same result.
There were differences though. The bath application of serotonin caused things that you never saw with GPR because it was so intense and for so long. And we saw in Tritonia, we saw differences with serotonin stimulating the DSIs. We had really inconsistent effects when we bath applied serotonin. It's because it didn't adjust so the neural stimulation was state and time dependent, and you couldn't get that when you were bath applying. So you lose the temporal component.
Right. And the temporal component is really, really critical, clearly.
Ron, so red herring or not, I mean, if you have a biological result that may be reductionist, but you have the states of the networks, right? And under control conditions and in the presence of dopamine. So if you hadn't defected 20 years ago and
10 years ago.
well, actually, yeah, that wasn't 20 years ago.
It was close to it, actually. I started working
well,
on...
you slowly, if you hadn't started slowly phasing out 20 years ago.
What's a decade?
But anyway, so if sort of like our ability to handle many versions of models and stuff like that, like would you expect that you could put in the whole laundry list of effects that you had described over decades and to a more useful degree than just fudging every single thing till the network model does what you want it to do? Do it in a way that you could test the general functional principles, at least. For example, your idea of the balancing opposing effects of presynaptic and postsynaptic dopamine action and things like that. Or is that just an exercise in, well, you get whatever whatever you fudged on all kinds of quantitative levels?
Yeah, well, my hope is certainly that it would show up. I mean, these results were all significant results, and I'd like to hear from Astrid about that, actually. I know one thing even I talked about right when Astrid was starting to come up with her results was, someone came, I can't remember who it was, came to my lab and we passed over the sum total of everything we knew about dopamine. And the idea is, could you make those changes and get the system to change the way dopamine gets it changed? And I don't remember that ever having been completed, but we did talk about it. It was, come on, I'm blanking out, a French fellow. I'm sorry, I've forgotten his name.
Jean-Marc?
Yeah, maybe. Astrid, what do you think about all this?
Oh, I don't have much to contribute.
Well, I mean, I think the problem is that since you have so much degeneracy in the multiple solution space of the control, that you're bound to get a lot even more difficulty if you try and put in all of the details. Because if you start out with a cell that has a larger calcium current, a smaller sodium current, or in a different case, the other way around, you're going to get different effects. So it might be very, very interesting, but it will take a little bit of cleverness to figure out how to do it right. Because
if I may say something here, I think if you don't account for the precise action of each modulator, of any of these modulators, on not just the effect on the amplitude, but the time courses and the midpoints of the voltage-gated channels, then it is impossible to predict what the outcome is because there's just so many of them. And the system is so highly nonlinear that you can pretty much get anything out of this if you just move things up and down and way around and just play with it. So if we want to predict things properly, we need to do it way, way more accurately at every single level. And this is a problem with which we run into. You need to do it all probably within the same animal, because there's so much variability from animal to animal that you can't take the average results and put it back together and say this is what it does, right? So, and we know this. It's impossible to actually say these components will lead to this result at the network level, even at the cell level.
Yes, and that's absolutely a totally central critical concept for all of neuroscience, this business that you can't use the average. That said, these effects of dopamine were totally reproducible. It didn't matter what combination that particular AB cell had, we still got the same result.
You got the same.
Absolutely.
You got the
Yeah.
same result qualitatively, but
Yeah,
that
absolutely. Or even you get the same change, yeah, but there is variability.
the minute you start putting that onto variable starting conditions, you're going to see, because there are 5 or 6 or 8 or 12 things happening, you're going to see it play itself out. Or well, maybe some of them will actually collapse down. Maybe there'll be sort of a reduction.
I'm not sure I entirely disagree, because I entirely agree, because the goal isn't really to rebuild the exact version of the STG in a computer. The goal would be to see if I put these principles back into an interacting set of variables, if you have a bunch of neurons and a bunch of synapses, and see if the general principles, for example, of opposing actions and things like that go into the direction, or what would it take to push them into the directions that you see in the dish. And then feel free to do this a bunch of times with different combinations to see that you're not just finding an idiosyncratic solution for a specific model, network model that you build, but it doesn't, I don't know, it makes things a little more complicated, but I wasn't talking about, you know, of capturing everything and building, you know, a completely accurate quantitative model of one specific STG in the computer. There's a lot to be learned before you attempt that, right?
Yeah.
Simply taking these actions and putting them together without knowing the answer of what the network does, I would claim that it's impossible to predict that with this much complexity and this much nonlinearity. And we're only talking about 6 types of neurons. I mean, people do this all the time in brain systems. And I, you know, having worked now for a year on these things, I would say that the systems they're looking at conceptually are so much simpler than what we look at. It's just conceptually, they make these claims because you know post hoc the answer. If you didn't know the answer, there's no way you can predict this, right?
Well, but they're also, you're just using 3 cell types that just have a 15,000 copies for each of the 3. So.
Yeah. Right, with the slight variability in the conductance of each one.
And also, may I?
After you.
Sorry to do that.
What? May I? Go ahead.
I was just going to suggest in the opposite direction is that, you know, if I understand what everyone's saying is that there's such a large parameter space that you won't be able to recapitulate what happens. But I was going to, I now feel stupid about my original question. I was going to say, why don't you dissect the receptor subtypes? Because I think the strategy that you've been implementing is really the right one, because you may find what's constraining the system. What if you find a general rule about the signal transduction mechanisms? Like you said, you know, there's only certain signal transduction mechanisms that you can elicit, you know, and somewhere these receptors have to converge. And the thing is with reductionism, it does seem to get more and more complicated, but I kind of like the approach, because what if there's an emergent property of the system where, yeah, it looks complicated now, but maybe we'll find a pattern as we reduce things. And I know it's kind of a blind faith in reductionism, but I kind of like the direction, because we don't know if we go up a level or go down a level, which one is the right way to go a priori. And I understand from a modeling perspective, it's going to be very difficult because you have all these free parameters. But if you find that this signal transduction mechanism always has this effect, I think that could be really informative, but I'm very highly biased.
Good point.
Pierre, you were going to say something.
Yes, no, I want to, I don't know, you hear me?
Yes.
Yes, you hear me. Yeah. What we have with the stomatogastric system when it's in the dish, in vitro, it expresses a lot of flexibility, we can go very precisely in the details. But if we look what is going on the animals, if you start to record at the animal levels, which is at the end, what we must look, the physiology is for the crabs, is for the lobster, is for the shrimp. And here, what we know they are not too much of work done on the behavior of this thing, but Maurice Moulin did, and Elisabeth Reiser, and also Stefan Clements did something. And even Stefan was able to record the animal for days and years, even for someone. And the system do not express such a flexibility, We have a system which is very stable, which is sometimes when you have a molt, you see big changes in the pattern, but nevertheless, it's quite very stable. It looks like when the stomatogastric nervous system is within the body, is trapped, is in cage, is expressing very little, indeed. So I don't know, maybe this huge flexibility that we see in the dish is maybe we must put this thing in a perspective of evolution, That we have a lot of different lobster, a lot of different crab, a lot of different shrimp, it's huge, this family of crustacean. And the stomatogastric seems quite stable. So therefore, body changes, but the brain, which controls this body, is not changing, but is ready for all the changes. So it's rather a potential for the unexpected thing, rather I would say to deal with a specific physiology in the given animal. I don't know if I am clear, but it's what I want to express.
Can I add something to that?
Sure.
Sorry?
So, I mean, this is of course very far removed, but from the stuff that I did in New Zealand, it looks very much like the somatogastric system is doing other things than what we think. Of course, it's mostly doing chewing and eating, but we found that it has a huge role in sound production, and that sound production actually is dependent on mating behavior. So the lobsters that we looked up and the crabs we looked at use sound, and they use the gastric mill to make sound and do that in mating situations, especially the males when they fight about females. So I think that the system needs to be much more flexible than just dealing with chewing mussels and maybe squid or something. So just a little background.
Yeah, that's a fantastic story, Mary. Really wonderful.
Your daughter, can we say hello?
Do you want to say hi? She's hiding now. Here she is.
Oh, that's cute. Paul, do you want to comment on the evolutionary variability?
Yes, so that you are saying that within and how you record these animals, because you seem to express that this stomatogastric system is have not only the function of chewing and so on, but is about to make what kind of communication it's what it will be.
So the crabs have that we looked at have 3 different sounds. Some of them seem to be just made in a state of excitement. So if somebody walks through the tank and they get and they know they're about to be fed, they start making these sounds. There's an other type of sound that we've only heard when a male has a female in hold position, and then other males approach and want to basically get to that female, they start making sounds. And so what we don't know is, this is really just a start, some preliminary results. We don't know the details of, do they only make those 3 sounds? Do they make more sounds?
And how this sound are produced?
I'm sorry? Oh, it's by grinding the gastric mill. So I did in vivo recordings of the gastric mill muscles and so EMGs while they were producing those behaviors. And so we saw that it was actually the gastric mill producing those sounds.
Okay, I will look with pleasure.
Do you see any behavioral impact of the sounds? I mean, does the female react to the sound at all?
The female doesn't react, but it doesn't have much choice once it's in this holding grip. It's really, it's kind of a mean, brutal mating ritual that they have.
Yeah.
But it does seem that if you play sounds, the males respond to the sounds and find that they.
Yeah, to me that to get any reaction, I think would be a necessary observation for me to believe that it's actually communication rather than an epiphenomenon of how the gastric mill moves.
Exactly, and that's quite tricky to figure out because at first we thought, oh, maybe they're just starting up the gastric mill because they're anticipating food, and all we're hearing is that the expectation of food because it's like stomach grumbling. But we actually do see behavioral, a lot of behavioral evidence.
Oh, it's good.
Really something.
Can I just use this pause to remind everybody that in 3 weeks from today, we have the SDG annual meeting. And I hope that as many of you as you can attend. And also November, we don't have a presentation of our monthly meetings, but in December, we have 2. On December 2nd, Mikey has graciously agreed to give a talk to all of us. And on December 14th, Ron Calabrese has graciously agreed to give a talk to all of us. So I hope that everybody can join. And also, I think Smita and Nelly sent out a request for how we should do the spring meetings and what we should do in terms of whether we want to go on specific topics, groups, etc. Thank you.
Thanks to everybody.
Right.
Thank you.
Actually, yeah, can I say, I really want to thank Ron for doing this, because as someone who has read most of these papers multiple times, including the review articles, I have to say this was such a great sort of grand overview of things that if I have a student now who wants to read themselves into all the amine modulation stuff, I will tell them to start with a recording of this presentation. So thank you very much.
Absolutely, I agree. And Farzan, you're going to send it to us, right?
Yes, on this note, I'm going to stop the recording and I will send the link through Smita, and Nelly will send it actually. Thank you.
Okay, great.